So what he did was to build what some of us call a crammer machine. It's all of about 24 inches in diameter. And it is a cage about 8 to 10 inches high and 24 inches, and it's made of fishnet. So the bird even bashing around it isn't going to get hurt. And here's your starling inside, roughly the same proportions. And around this are placed little feeding boxes at compass points. We don't have to bother with all of them, you get the idea. The box has a latex sheet in front of it. The bird can't see into it. But you put food, some kind of bird seed, in one of the boxes. And the bird very rapidly discovers that the food is in this box. When it's put in at 6 o'clock in the morning, this is north, this is south, west, and east. And I warn you, you're going to get misled in the first experiment I tell you about. Cramer was misled. It's fun. He trained the birds to go to the east at 6 o'clock in the morning. And after a couple of days, you put that bird in there at 6 in the morning and boom, it goes straight to this box. It knows where the food reward is. So the question is, has it learned that it must use the sun as a compass? And part of the proposition is that if that's the case, it must have a clock, because the sun is constantly moving at 15 degrees of arc per hour. So the first experiment was to bring the bird in at 9 a.m., 3 hours later, and expect the bird still to go east, knowing where east is. It came here. At 12 o'clock, with the sun south, it came here. And that bird, you see, is not very good. It was, as is said, retired from experiments. And it was kept, though, as a pet and given the name heliotrope. It went to the sun. And the joking version is, of course, that when the bird first learns this, it says to itself, I mean, you understand I'm playing games now, I wonder if these turkeys know that I have a good clock and an almanac and know all about that. The simplest solution, says the bird, is to go to the sun at 6 o'clock in the morning. And that's what it continued to do. The next starling brought in here is trained to go east, but at 9 o'clock. So there's no confounding the position of the sun now being exactly where the food is. You have to learn, the bird has to learn that the food is 45 degrees to the left of the sun. And it does. This bird now, when tomorrow you bring it back in here at noon and the sun has moved from there to there, it still goes to the southeast. It still goes when the sun's here, and in fact, all through. It looks good. But the really beautiful experiments which were then done was to take this whole apparatus, this crammer machine, indoors and put it inside of now a big nylon tent. Here is the table with the crammer machine on it, and the food box is here and the bird's inside. Inside a laboratory with windows closed, blinds down, and you put a 500-watt lamp here, which acts as an artificial sun, a big disk of light on the nylon tent. And you hope that the bird is going to take the artificial sun as the real sun. So you train the bird to go east. And now you bring it in to this artificial universe. And at 6 a.m. the bird goes here. You don't give it too much. 7 o'clock you bring it in, and it goes here, here, at the next hour. 6 hours later it's going due north. All through the day, with the sun held constant here, the artificial sun. The bird keeps adding 15 degrees of arc to the direction it goes in relative to the sun. And in fact, if you keep the bird going, which they did, it'll make a 360-degree circuit. What comes out of this experiment? First, the bird is using this artificial sun as a direction giver, a compass, if you will. It knows, quote unquote, whatever that means for the bird, it knows, it's informed, that the sun's azimuth position, the azimuth position of the sun, is where on the horizon the sun would be if you drop a plumb line from it. In the summer solstice, it arises due east at 6 in the morning, and it moves 15 degrees of arc per hour and completes 180 degrees in the course of the day, setting in the west. The bird knows that the azimuth position of the sun moves at 15 degrees of arc per hour. It is assuming that this lamp here is the sun and that the sun is moving in that direction. Incidentally, the bird knows it's in the north hemisphere, not the southern hemisphere, which would be the opposite direction. The bird knows it's in the northern hemisphere and that the azimuth is moving at 15 degrees of arc per hour. And the final thing, it must have a clock. It can compensate for the passage of time. Well, you can imagine how excited I was in hearing all of this, and went home to think about it. Before, what happens after that in Kramer's laboratory and some of the things I became interested in, let me point out, I've told you what this experiment does tell you, and I overstated it. I said the bird has a clock. You don't know that. You know the bird has access to a clock. Something's telling it the time. And you don't, at this point, know the clock's inside the bird's brain. This is where, as biologists, you have to be careful. And you have to go with Mr. Popper, Karl Popper. All you know is the bird compensates for time. Whether the clock it uses is inside or outside, another question. And let me go ahead to settle this issue. In 1950, there was no proof that the clock was inside the bird. But everybody, Kramer and the audience, that's me at the moment listening, convinced it's inside the bird. The proof came about 6 years later from one of Kramer's students, a man called Hoffmann, Hoffmann, Klaus Hoffmann. And as you're going to see, the Kramer group and my own laboratory by 56 were pursuing the guess that Bunning and Calmus's rhythms reflected the presence of a clock too. The bean plant, in some sense, knows 24 hours. What it's doing is determined to a near 24-hour periodicity. These insects can measure time. We're going to come back to that. What was known about the Bunning-type, Demiran-type rhythms was that they're brought into phase with local time, so that in the case of the insects, this happens near dawn, for instance. Brought into time with local time by virtue of their response to the light-dark cycle. To oversimplify, and this is wrong, but it's a useful, simple way of putting it, they use sunup as a reference as to what the time is. Now, The question is the clock the birds have inside them or outside of them? Is it Rosa Stoppel or is it Demiran? Some oscillating system inside, some periodic system measuring time. Well, you know that Demiran rhythms are brought into phase with local time by the light-dark cycle. So Klaus Hoffmann took birds, same institute, same equipment, same starlings, trains them to go east in a Kramer machine. They're in pursuit of food. And he then, that's outside, brings them into the laboratory and exposes them to an artificial light-dark cycle in which dawn is brought up 6 hours ahead of the outside world. So it's brought up midnight, local Wilhelmshaven time, instead of 6 a.m. If it were this, I don't know what time of year it was done, but it's advanced 6 hours. And the birds are exposed to this phase-advanced clock, this phase-advanced light cycle for a week. And then he takes them and brings them outside and says to the bird, now go find your food. What do you think happens? Anybody got a guess as to what happens? Now, what are we talking about? One possibility is that the birds read Rosa Stoppel, that is to say, they read factor X. They're being driven by some physical cycle, change in the Earth's magnetic field, we don't know what. That's how they know the time. If that is the case, if that's their clock, the switched light cycle has no impact on them. And when they come out, and oh, by the way, you've already shown that the birds will remember for 6 days or so. They'll go east, which is where they were, quote unquote, trained, where they learned the food was. If, on the other hand, the clock is a periodic system of the Demiran rhythm burning type, you know that its exact phasing is established by the light-dark cycle, and you've brought dawn up 6 hours early. So if you bring this bird out at, let's say, 6 p.m., it would normally go here, I mean, east. Have I done it? Yeah, but it's only noon. So what will happen? It makes a 90-degree error, which is what you predict on the hypothesis that you've reset the phase of the clock. It thinks, if you bring it out at 6 p.m., it ain't 6 p.m. He looked at his watch. And his watch says, no, it's only noon. So where's the sun at noon? It's here. And so east is here. I'm blowing this. He makes a 90-degree error this way. Now let me finish that. I'm getting a little worn out here. I have to warn you, I've got some kind of a grip or something. Have to hurry. A few years later, an American student, Kenneth Rawson, from Swarthmore, working in Kramer's lab, began an experiment which the first time around didn't work very well. It was picked up and repeated by a German, Schmidt-Koenig, names aren't important. And what Schmidt-Koenig did is absolutely marvelous, and you must hear about, even though you don't need it at this point. Someone in Kramer's lab, a lady Ursula St. Paul, had shown that homing pigeons, when they home from some distance, contrary to Army Signal Corps manuals, forget it all. They don't have to be trained or learn. If you take, as these people did, that is to say, Rawson and later Schmidt-Koenig, if you take, that's by the way Denmark, you see. I'm sure you would have recognized it. And there's Sweden. And Wilhelmshaven, where Kramer's lab is roughly there. And down here in Westphalia, there's a town, Gießen. It's about, as I recall, 200 kilometers, something like that. If you take pigeons, homing pigeons, that have never been out of Wilhelmshaven, and take them down to Gießen in a black box, real black box, and release them from the top of a tower, you can record their initial headings. The control experiment. All the birds go home, or rather, to be exact, they're pretty good. There's a certain amount of slop. Occasionally a bird finishes up in Copenhagen and knows it's blown it because after a night's sleep it goes straight back to Wilhelmshaven here. That's a fact. But at any rate, the control birds head with a very small variance on their direction home. If you take these birds and subject them to a Klaus Hoffmann, keep them in the lab under an artificial light-dark cycle, phase advanced by 6 hours for a week, take them back to Gießen, they don't go to Wilhelmshaven, they're off to Amsterdam for a party, obviously. Now, that's a slight oversimplification. It isn't in the sense of initial headings. Most of them eventually finish back in Wilhelmshaven. Days later, they catch on somewhere along the line that they're blowing it. But the initial heading is predictable from the extent to which you reset the clock. All right, enough of all that. Kramer and his school. And at the same time, von Frisch's school, but with confused language in their description that misled him. I wish I had more time to tell you what that meant and did. Beside the point here. 1950s. Birds and honeybees have got good clocks that are modulo, as one says, 24 hours, 24-hour clocks, roughly. They're internal to them. They run on metabolic energy. You can stop them in helium, etc. Question, what is the clock? I'm back to 1950, before Hoffmann's experiments. And I went home and thought, and this is going to be almost the final section to this, supposing, and I can't remember now, and I've been trying to recently, was it 1, 2, or 3 days after hearing Kramer, that it occurred to me that Bunning's rhythms, I was groping for something like this, you know, Bunning's rhythms, de Myran's rhythms, the birds or the insects involved are doing something with a 24-hour period. There's another way of saying that. Those organisms can measure the passage of time somehow or other. There's something in the way they're built that measures time. That reformulation of the observation was almost as exciting to me as listening to Kramer. That may seem silly to you, but there's a general point. It's how you look at an observation and how you describe it. I'm not sure I shouldn't tell the time and tell you how the von Frisch people working with bees described their observation in not only an unprofitable way, but a distracting way. They referred to the bees having time memory, Zeitgedächtnis auf Deutsch. And a few years, in '59, I visited Munich and talked to the Frisch people and said, why have you still not reset the bees' clock with light, the way all these people have in birds? And the person, whose name I won't mention, it wasn't Frisch himself, but his leading student said, no, bees don't have clocks, they have Zeitgedächtnis. And the notion that it's time memory, that word doesn't have a connotation to it the same as the word clock. Your wristwatch is no good to you unless you can set it to local time. And by not using the word clock, Kramer's group did Uhr auf Deutsch. As soon as you use the word clock, you're reminded the clock's no damn good to you unless you can set it to local time. The Frisch school missed that for years. So watch what you say is the message. All right. The excitement for me was in redescribing the observation of a persisting rhythm to imply that the plant could measure time. In fact, when you're done with it, of course, dummy, did you ever doubt that? Never occurred to me before. All right, now if it's a clock that's inside these insects, and it's the same clock that's used in bird navigation, there's one thing you can say about it. The rate at which it moves must not vary significantly with change in environmental variables, or it's a lousy clock. You know, well, I doubt you do know. I didn't know until somebody pointed it out to me, that in the history of chronometers, the technology in the Western world of chronometers used by navigators, it was James II in England, whose merchant fleet was important to him. They needed a really good chronometer in their navigations, and the challenge was in compensating existing chronometers for their susceptibility to change with temperature. And I'm sure you all know about how you compensate for the period of a pendulum moving with temperature. You use two metals. The period of the oscillation depends on the length of the pendulum. You've got to keep that constant. What about Bunning's rhythms? They're endogenous. They use metabolic energy, so Chalmers said. Chalmers showed. If they're metabolic, everything about metabolism that we know about, that we knew about, there's a little exception to that, but nearly everything in metabolism, the rate of it varies with temperature. The chemical reactions in the cell, and when you raise the temperature, they go faster. When you lower the temperature, they go slower. If Bunning and Chalmers are correct, that the period of the rhythm is lengthened, going out to 36 hours by dropping the temperature, forget it. That rhythm's no good as a clock. It'll misinform the bees and the birds. So I decided to pursue this issue, and if I had more time, I would, I think, amuse you by telling you how in our try, very quickly, the first experiment was done. I decided to repeat Bunning's experiment. It was very simple. This is in 1952, it so happens. It's a long time ago. And I want you to remember, and I'll draw it. These are our Drosophila doing something once per about a day. And at this point, Bunning had dropped the temperature from 26 degrees to 16. And we're going to call these peaks 1, 2, and 3. And normally the period between the successive peaks is about 24 hours. When he dropped the temperature, peak 2 comes out. Here, 2 star, 12 hours late. The period of the rhythm is apparently temperature dependent. No good. I'm going to find out if that's the case. And I wanted to go to Woods Hole, you may have heard of, but I found that to spend the summer in Woods Hole would cost me 33% of my annual salary, which was then $4,000. So we didn't go to Woods Hole. We went to a place in the Rockies called the Rocky Mountain Biological Lab at 11,000 feet. And one of the outcomes of that summer was a lifelong love affair with the Rockies. Marvelous. But when I got there, the station had no temperature controls at all. However, I found that about 5 miles up the road, a little over 11,000 feet, there was an abandoned mine and an abandoned outhouse in it, a real good old one-holer, which was so old, it was odorless, and with just some tar paper and galvanized nails, I could make it into a little dark room. It's you didn't need an NIH grant for this. And the point is that right next to the outhouse was a marvelous little creek running off from an ice field way back. So the temperature was, I didn't have a thermometer, but it was damn cold. And the trout loved it. So I could go up and fish and do an experiment. And I had taken Drosophila with me, and I had one set of cultures going in the outhouse. By the way, I did put some clean plyboard down on the seat so it was touchable, do you see, as a workbench. And here's the outhouse, dum-dum, and outside's the creek, with trout in it, which I caught. And my wife, knowing we were going to 10,000 feet, had brought an old-fashioned pressure cooker. You don't see them anymore, about this big. They're watertight when you tie the lid on, and to the outlet on the lid, pressure cooker looks like this, do you see? And this is a watertight seal on the lid, and the steam outlet here, you put a black rubber tubing on it as a snorkel. And where's the creek? Oh, it's here. You can put the pressure cooker in the creek with a rubber snorkel so that the thing gets air and doesn't get water. And I put in cultures of flies into the outhouse and some inside the pressure cooker. And, you know, it's a messy experiment, but I left them 2 days and came back, and on the second day, using the outhouse as a dark room, I could close the door to find out how many flies had emerged since the last observation an hour ago. I could measure the rhythm. Inside the outhouse, it was, well, this you see, wait a minute, where am I? Inside the outhouse, it was like this. This is the old control, and this is the outhouse. Essentially, the details I'm not going to bother you with. Essentially the same as that. In the stream, you see, I'm not observing here. I'm going there. In the stream, like this. And no, I'm sorry, I've blown it. It was, I'm sorry, I'm tired. The first peak, the second peak in the stream is on time, like this is. And, you know, what Bunning and Chalmers had reported was that the peak was delayed. What they had looked at was peak number 2. It was delayed. I didn't look at peak number 2. I thought this is a simple preliminary experiment. If there's a difference between the outhouse and the river, it's going to be amplified in 2 cycles. So I looked in the 2 cycles, and there was a difference. But it was tiny. It was about an hour and a half after 2 days. In other words, whatever was controlling, you've got to stay with me because there's a fun story here. Whatever was controlling the fly's rhythm was going almost as fast at about 10 degrees, the stream, versus the 25 or so outside. Very little effective temperature. But then what was this story of Bunning's? That the peak was delayed. But at any rate, I remember going home that evening, very excited. Everybody who does that research knows when they've got a bear by the tail. And I had a bear by the tail. Went back to Princeton, where I was working, and decided we're going to repeat this experiment with good equipment. And did. And went through this whole schmear, 26 to 16. And I had 2 undergraduates helping me collect the data by hand in those days, every hour on the hour, looking at about 100 vials and collecting flies. A lot of work. Well, when we dropped the temperature, unlike Colorado, which is this experiment, when I didn't look here, we found that this peak was delayed 12 hours, just as Bunning had said. And then, instead of going home excited and feeling I had a bear by the tail, I was very glum. Went home. I remember the weight was all timed. It was about 11 at night. Went to bed after a little liquid refreshment. And went back into the lab next morning, and the undergraduates had stayed up all night. And they came to me absolutely perplexed and said, look, the peak's here. This is peak number 3 now. It's only about an hour and a half late. That's what happened in Colorado. And what I hadn't done in Colorado was to look at this peak. Now, this is only a beginning lecture, and I'm not going to go into details. I have spent years clearing up the mess. The story is that after this peak, every other period is 24 and a few minutes long. The effective temperature is trivial. What the experiment showed in Princeton